Welcome to the video lectures for this week. We'll be looking at Chapter 17, which was the immunodeficiencies, autoimmune diseases, hypersensitivities, and vaccines. So mainly the problems of the immune system and then how we can use the immune system to benefit us. And then we'll also look at microbial pathogenesis. All right, so remember, these were your outcomes for Chapter 17. Let's go ahead and walk through some questions. This part of 17 really is just about understanding definitions, so being able to tell the difference between an autoimmune disease and an immunodeficiency, or an immunodeficiency and a hypersensitivity. So what do those words actually mean? So we can look at this question here, which is going to ask us which one of these diseases could be the disease that Zach was diagnosed with, because he was just diagnosed with an autoimmune disease. So if we read through those, we should be able to identify which one is describing an autoimmune disease. A says we have a peanut allergy because of IgE. B is type 1 diabetes, where the cytotoxic T cells were attacking cells of the pancreas. C was SCID, where there was a genetic mutation that prevents them from forming adaptive immune cells. And then D was HIV, where it was a viral infection that lowered the number of functioning cells. So an autoimmune disease is when your immune system is no longer recognizing the difference between self and foreign antigen, and so now it's attacking you. And so when we look at those, the correct answer to this one should have been B. Type 1 diabetes is an example of an autoimmune disease because your own cytotoxic T cells attack your pancreas cells as opposed to attacking something foreign. A here, this is an example of a hypersensitivity because the immune system is overreacting to something. So a peanut allergy, if you've got a peanut antigen, that is foreign, but it's not something you necessarily wanted the immune system to really be attacking. And then C and D are both immunodeficiencies, where there's some sort of problem with the way the immune system can function and develop. But C is what we call a primary immunodeficiency because it was there from birth. There was a genetic mutation they were born with. And D would be an acquired or a secondary immunodeficiency because this is something they developed later in life, in this case because of an HIV infection. All right, so now let's look at this next question. Besides just being able to tell the difference between what an autoimmune disease and, say, an immunodeficiency is, we should also be able to tell the difference between the 4 different classifications of hypersensitivities. So our problem here is going to go through and say, okay, surface of your red blood cells has a protein called the Rh factor. That's actually the plus when you hear about someone saying they are A positive. The positive is actually an Rh factor. And so if you're A negative, you don't have the Rh factor. So for an example, an Rh negative mother is giving birth to a second Rh positive child. And because she's Rh negative, she has antibodies against the Rh positive, against that protein. And those IgG antibodies can cross the placenta, bind to the fetus's cells, and that activates complement, which would harm the fetus. And we want to know what type of hypersensitivity this is. So let's look at the 4 types of hypersensitivity. Remember, type 1 involved IgE, and that's where IgE bound to the mast cells, and then later, when that IgE binds to its antigen, it signaled those mast cells to release things like, not mast cells, basophils, to release histamine, and then that histamine causes the effects of an allergy. Type 2 hypersensitivities is when you have antibody that's going to bind to some sort of antigen on the surface of a cell, and then that activates complement on that surface. Type 3 is when you have antibody that binds to antigen, but it's soluble antigen, and that forms these big clusters that get stuck in the tissue and then activates complement. So both 2 and 3 end up causing damage to cells and tissues because of that complement activation, but they activate it slightly differently because of where the antibody has bound. And then type 4 is the only one that didn't deal with antibodies. This is mediated by those cytotoxic T cells. So when we look at those descriptions and we go up to the problem, we can see that we've had an antibody, its activated complement, so it's either going to be B or C, and if we look to see where that antibody was binding, it was binding to the fetus's cells. And so that's a cell surface, so this would be type D. And what that ends up looking like is you've got a red blood cell. On that red blood cell, you have the Rh protein, and you have antibodies from mom that bind to that Rh protein, which activates complement. Okay, remember, we activate that whole pathway. Eventually, C9 comes in, pokes holes, and actually harms the fetus's cells themselves. Okay, so those are kind of when the immune system goes wrong. This chapter also had information on doing the vaccines and what vaccines are, so that's what the next couple questions are going to look at. Okay, so we're developing a novel vaccine, and we're going to use a mutated form of the pathogen, which is no longer pathogenic. So we don't know what type of vaccine this is. So again, a lot of this chapter is terminology, just understanding what the different types of vaccines actually are. Okay. An attenuated vaccine is when the pathogen's alive, but it's now non-pathogenic. So either it's been mutated, so it's no longer a pathogen, or sometimes you can use a weakened form of the pathogen, for example, a pathogen that might infect a different species. And that, you should see, describes the same vaccine that's in this problem. So a mutated form of the pathogen that's no longer pathogenic. These other ones, so inactivated, is when you take the pathogen and you kill it. And so this is a dead organism. It can't replicate, it can't divide in the body. Subunit vaccines, this is when you just take a portion, typically a protein, because remember, those are more immunogenic. And then you just use that protein as the vaccine itself. And so, for example, something like coronavirus that has those spike proteins on its surface, we can take just that spike protein and use that as the vaccine, and that would be an example of a subunit vaccine. Toxin vaccines are also very similar, only that protein would be a toxin, so you actually have to inactivate it first before you use it. And then the really new vaccines are these DNA vaccines, which is instead of taking the protein itself and using it for a vaccine, you actually go a step further in the central dogma and you take the DNA itself that would have been transcribed and translated into that protein, and now we use just the DNA as the vaccine. And when it gets into the body, it's transcribed and translated into this foreign protein, which ends up activating the immune system. And so those are the main types of vaccines that currently exist. The attenuated vaccines are the ones that tend to have the best immune response because they are still alive. They can replicate in the body, and your body sees them more as a true pathogen than some of these other ones. But because they're alive, they do also have negative side effects where you do have to, grow them and store them properly because they're living cells. You also sometimes can have problems if they mutate back and become more pathogenic. That's always going to be a concern. Okay, so let's look at this next question, which is really talking about the whole purpose of vaccination. Okay, so it says, Tyrone is a 5-year-old boy with leukemia. Due to his cancer, he's unable to be vaccinated against measles. He lives in a community where 95% of the children are vaccinated. So what does this mean for Tyrone? Since he can't be vaccinated, there's a good chance he will contract measles because his community has herd immunity. Since he can't be vaccinated, there's a good chance he will contract measles since there is no herd immunity in his community. Or he's in luck, his community has herd immunity, so he should also be protected from measles even though he cannot be vaccinated. And this is the answer to this question. So remember, herd immunity is what happens when a high enough percentage of the population is protected through vaccination. So if one person becomes ill, there's no place for that pathogen to spread to. It can't spread through the population because everyone else is immune to it. And that's what herd immunity is, and that's one of the main purposes of vaccinating at high rates, is not just so that you're protected, but so that the population as a whole is protected to help protect those that can't be vaccinated for some reason. Okay, so now we're going to move on to the microbial pathogenesis section. This section contains information for both chapters 2 and 18. Mainly the information in 2 is an introduction to the information that's in chapter 18 and the information that's in chapter 26, which you'll do next week. And so some of these you can see with asterisks on them in chapter 2 for the learning outcomes really tie more to the things we do next week in chapter 26 as opposed to chapter 18. Okay, so both the sets of learning outcomes are here, but they're really put together in one set of lectures. Okay, so we'll look at this first question, which is going to talk about the ID 50. And so we have to go over and understand what that is. So we're working with 2 different bacterial pathogens in the lab. Pathogen 1 has an ID 50 of 10 organisms, while pathogen 2 has an ID 50 of 10,000 organisms. And the question says, which do we need to be more careful when handling? So first we need to stop and think about what was the ID 50. So what does that term actually mean? And let's see, I'm going to stop drawing here for a second. Give myself a little bit of room to write. Okay, so if you remember, the ID 50 is down here we're going to have the dose, which is going to be the number of organisms. And over here, we're going to have the percent of the population that is infected. And when you look at these, for every type of species, a pathogen, there's going to be a curve that you're going to see. It's going to go something like this. And the ID 50 is whatever dose, so whatever number of organisms you need to actually cause an infection in 50% of the population. And so in this problem, we've got 2 pathogens. One had an ID 50 of 10, and one has an ID 50 of 10,000. So we'll say this would have been pathogen 2. Down here is our 10,000. An ID 50 of 10 would be something like this, where we've got our 50% here. So this, say, would be pathogen 1. And it wants to know what do we need to be more careful when handling. So remember, the ID 50 is telling you how infectious something is. The lower the number, the fewer number of organisms you actually have to be exposed to get the disease, which means the more infectious the pathogen is. So the lower the ID 50, the more infectious the pathogen. As we look at these, a pathogen 1 clearly has a lower ID 50. That means it's going to be more infectious, and when you're working with them and handling them, you have to be much more careful handling that one because only a few organisms can make you sick. And so if we read through the answers, the answer to this one is going to be B, that pathogen 1, because the lower the ID 50, the easier it is for that organism to infect you. Okay, the other part of what we call pathogenicity is virulence. So pathogenicity, or the ability of an organism to be a pathogen, really consists of 2 different traits. The infectivity that we just talked about with the ID 50, and virulence, which we're going to look at now with the LD 50. So the LD 50 is the same concept as the ID 50, but instead of being the infectious dose, it's the lethal dose. So how many organisms do you need to be in the body to actually be lethal to an organism? So we're going to read this problem here. It first says we're trying to identify virulence factors. Remember, a virulence factor is just anything that the pathogen has that allows it to be a pathogen. Okay, there's 5 main steps of microbial pathogenesis. And remember, you are going to have to enter the body, attach, and colonize. And in order to colonize, you have to evade the immune system. You have to damage tissue, and then you have to exit to transmit. Any of the microbial factors that allow those steps to happen are considered virulence factors. And so in this problem, we're trying to identify what some of those virulence factors are for this pathogen. We delete a specific gene, and find that the LD50 changes from what was 1,000 organisms to now 1,000,000 organisms. So we want to know if the protein that was encoded by that gene is a virulence factor. Remember, virulence factors are anything that's going to affect its ability to be a pathogen. We just changed the LD50, which means when we deleted this gene, we took an organism that had pretty high virulence. 1,000 organisms to kill 50% of the population is relatively high virulence. To something that was much lower virulence. Now it takes 1,000,000 organisms to kill an organism. And so that change in virulence says that yes, whatever we deleted was actually a virulence factor. It was involved in making this organism a pathogen. And so our answers can't be A or B because those both say no. C says yes, a virulence factor is anything the pathogen needs to cause disease. When you removed this gene, the organism now shows lower virulence, meaning the gene played a role in pathogenicity. So C is going to be the answer to this one. Remember, we weren't necessarily talking about infectivity if we compare the answers in C and D, because we were looking at the LD50, which tells us about the virulence, so how deadly a disease is, not necessarily the infectivity or how easily it is to infect something. You can have really low virulence, but still be highly infectious. Something like the common cold is easy to spread, it's relatively infectious, but it has very low virulence, and so they don't have to be tied together. Okay, so remember the steps of pathogenesis? The organism would have to enter the body, it would have to attach to tissues, it's going to have to avoid the immune system to actually grow and replicate, and it's also going to be causing damage. And so this question is going to talk about how pathogens can cause damage. So we've got a bacterial pathogen is causing tissue damage by secreting a toxic protein directly from its cytoplasm into the cytoplasm of the host cell. And it says, okay, this pathogen is using something secreted by something as part of its mechanism of pathogenicity. So we're talking here about a toxic protein. That tells us that this is going to be an exotoxin. So pathogens can produce exotoxins or exoenzymes. The difference between those is exotoxins are a toxic protein that has a specific cellular target. So it's going to bind to a certain type of cell and interact with it in some way, as opposed to an exoenzyme, which is a little bit more generic. An enzyme just has enzymatic properties. It gets secreted from the bacterial cell and goes and chews up stuff. So maybe it's an enzyme against phospholipids, and so it goes out and it degrades phospholipids of cell membranes. But it's doing that against all phospholipids in all cell membranes, as opposed to an exotoxin that, say, is just a nerve toxin, is only going to interact with your nerve cells. Much higher specificity. And those are both different than endotoxins, which, remember, is just another word for LPS. So endotoxin is an actual part of gram-negative cells that just sets off a big cytokine storm if it gets in the body, and it's really not anything that the pathogen itself is trying to do. And so we know this one's going to be either A or D, because we've got an exotoxin secreted by some mechanism. And the options that they gave us were Sec-A or a type 3 secretion system. Sec-A is the standard secretion system that is going to get you from the cytoplasm out into the external environment. If you're a gram-positive cell, remember you've got a thick peptidoglycan layer out here, your Sec-A is really all you need to take a protein that's inside the bacterial cytoplasm and secrete it out into the environment where it can diffuse away to go do its function. Whether that's an exoenzyme or an exotoxin. But if you're a gram-negative cell, you've got a cell membrane, and then you also have another outer membrane. And so in this case, Sec-A can take that protein and secrete it across the first membrane, but now it's trapped in the periplasm, and so it's going to need another secretion system to get it across the outer membrane. These are typically called type number secretion systems. So we've got type 2 secretion systems, type 3, type 4, so on and so forth. The only two you really needed to know were type 2 and type 3. So type 2 was a way to take this protein here that just got secreted by Sec-A and then use it like a plunger and push it the rest of the way out of the cell so now it can diffuse off to function. Type 3 secretion systems were the special ones that actually would connect the bacterial cell here to a host cell. So this would be a human cell. And it takes the protein and it actually shoots it like going straight through a hypodermic needle into the cytoplasm of the host cell. And if we come back to the actual description here, that's what's being described. So this is being secreted directly from its cytoplasm into the cytoplasm of the host cell. That is what the role of the type 3 secretion system is, so the answer to this one is going to be D. The benefit of this is that if you're using, say, Sec-A here, or this type 2 secretion system, if you want the protein to bind to this host cell, there's nothing that says it's actually going to do that. It could actually diffuse the wrong way. For your type 3 secretion systems, you don't have that problem anymore because you injected your toxic protein right from one cell directly into the other one. There's no chance of it diffusing away. So this is more efficient. Okay, so now we're going to look at kind of how we classify. Again, this is more terminology, looking at ways that these are going to be transmitted. So once a pathogen's been in a body, it's replicated and it's going to transmit to someone else. How can that actually happen? Okay, so we've got a species of virus here that can be found circulating in bat populations in nature. However, this virus has now infected human populations, where it's most often spread through contact with inanimate objects like doorknobs and tabletops. So how are we going to describe this pathogen? So A says the reservoir of the pathogen is the soil, and it's transferred through a common vehicle. B is the reservoir of this pathogen is other humans, and it's transferred through a common vehicle. C is this is a zoonotic disease based on its reservoir, and it's transferred through fomite transmission. And D is this is a zoonotic disease and it's transmitted through vector transmission. So the answer to this one is going to be C. Okay. This is a zoonotic disease because it's typically found in nature in bat populations. Okay. Zoonotic diseases are ones that have jumped from an animal into a human. And remember, the reservoir is just where it's naturally found. And so this is a virus that naturally lives in bats, but it has now jumped to humans. You can also have pathogens that, say, naturally live in the water supply, so their reservoir is the environment. And you have other pathogens that are only found in humans, and so their reservoir is actually the human body itself. And then we want to know about transmission. Hey, this is fomite transmission, and this would be considered indirect. Hey, fomites are just the scientific name for inanimate objects. So the doorknob is a fomite, and this is indirect contact because you didn't actually need to see the person leave, say, a pathogen behind. If you have pathogen on your hand and you open the door, you leave. 2 hours later, someone touches that same doorknob and they can pick up that pathogen. If we look at the other kind of options here, common vehicle is also indirect. But vehicles are things that everyone would have to have been exposed to get the pathogen. So this is typically foodborne diseases or waterborne diseases where you would have had to, say, consume the contaminated water. This is also an airborne disease, so the air itself can be a vehicle. But keep in mind, this is different than droplet transmission. A droplet is more direct contact because you have to physically see the person. You typically don't go more than 6 feet in droplet transmission, as opposed to airborne, which is actually circulating through the air. The other transmission mechanism that was here was vector transmission. And this is indirect because you don't need to directly see the person, but a vector is an insect of some sort. So this is transmission typically through things like mosquitoes or ticks. All right, and then the last question is asking, what is one key thing that a pathogen has to do to successfully colonize a host? Okay. So we read, hey, it must secrete a toxin in order to damage the host tissue. It must have a mechanism to evade the immune system responses. It must have a mechanism to attach to human cells or tissues, or more than one of the above are needed. So the answer to this one is D. But let's look at to see which ones of the above it has to do. So A says it must secrete a toxin in order to damage host tissues. So the key with this one is it doesn't actually have to secrete a toxin. We do have to damage host tissues in a way. Okay, so disease is the body not functioning properly. Infectious diseases is that malfunction is being caused by the pathogen. So in pathogens, they do have to damage the tissue, but sometimes they can do that just by growing in the body. Sometimes they do that by secreting an exoenzyme. They don't actually have to secrete a toxin itself, and a lot of pathogens don't. And so while they do have to damage host tissue, they don't have to secrete a toxin. That's one way that tissue can be damaged. They do, however, have to have a mechanism to evade the immune system. And so this can be done in a couple different ways. You can either have intracellular pathogens that try to hide inside cells, so they try to avoid the immune system that way. To do this, they're going to need to avoid the lysosome destruction. Remember, they could do that in a couple ways. They can either destroy the phagosome and just live in the cytoplasm. They can prevent the phagosome from fusing with the lysosome, so they never experience that lysosome. Or a very few handful of them have ways to detoxify the lysosome, so after fusion they just live in that environment without it killing them. You also have ways that they can disguise themselves. So this way they're trying to not look like a pathogen. One of the key ways they can do that is by having a capsule. So a capsule, if you're a bacterial cell, and here's all those antigens on your surface. Remember, these are those PAMPs that they're going to recognize. But if you cover all of those with a capsule layer, now you've disguised them and you can't see them, and that capsule helps you prevent it from being phagocytized. The other way they can disguise themselves is by changing their antigens, and viruses will do this a lot, where they'll mutate so the antigen on the surface doesn't quite look the same anymore, and so they avoid the memory response. And then the third one is to kill the immune system cells before the immune system has a chance to kill it. So there are a lot of pathogens that produce molecules that will say kill a macrophage. That way the macrophage can't phagocytose it. Or they might infect the adaptive immune system cells, something like HIV, so that they kill the immune cells before they have a chance to kill them. Okay, so this is the key with pathogenesis, is every pathogen, no matter what type, has to be able to do this. They have to be able to get into the body. Remember, they have to be able to attach to the cell so that they don't get flushed away. Then they have to colonize, and in order to colonize, they have to be able to evade the immune system, even if it's just temporarily. So they just need to grow to high enough numbers so they can transmit. And that most people are not dying of every infectious disease they get. Oftentimes your immune system will win in the long run, but on the short term, if the bacteria or the viruses or those fungi outcompete the immune system, they will eventually cause damage to the tissues. That damage leads to disease. And then the fifth step is the exit and that transmission mechanism. So that's everything about kind of chapter 17, where we had the immune system problems and vaccines, and then chapters 2 and 18, which is looking at microbial pathogenesis. If anyone has any questions on these topics, please do shoot me an email or reach out on the discussion boards. You can also sign up for office hours either this afternoon or any of the case study office hours for tomorrow you can use, because people may or may not be doing the case study yet since we haven't actually covered everything from next week. Okay, everybody have a great weekend.